Hello, I'm Lucia Gambuza with Modern Retina. I'm here with Dr. Ambati, Director of the Center for Advanced Vision Science, Professor of Ophthalmology at the University of Virginia, and co-founder of Inflammasome Therapeutics. Dr. Ambati presented 6-month phase 2 results for K8, a dual inflammasome inhibitor for geographic atrophy at ASRS last week. Dr. Ambati, thank you for joining me.
Thank you very much for the opportunity to share these findings with your audience.
So I understand you had some encouraging results. Walk me through what you saw at 6 months. What did the lesion growth data show, and was there a functional signal alongside it?
Yes, this was a 6-month multi-centered US-based phase 2 study of K8 sustained release implants in patients with bilateral geographic atrophy. And we found two sets of spectacular data. The zero point 0.7 milligram dose had a reduction in mean GA growth rate of 54%, according to the FDA mandated slope models of GA growth. The other two doses also had substantial reductions in GA growth rates, substantially higher than the two approved products, which in their first 6 months had reductions of 12.5 and 13.6%. In addition to the structural efficacy, we also found that overall there was a four-letter ETDRS visual benefit with the K8 treated group compared to the control group. That was also statistically significant. So this is perhaps the first time in a phase 2 study, certainly within the first 6 months, that we have seen both structural and functional benefit. And most importantly, beyond all that, there were no safety signals of note, no choroidal neovascularization, endophthalmitis, vasculitis, or intraocular inflammation. So all in all, very encouraging data that make us look towards phase 3.
Thank you for sharing those results. Did the treatment effect look the same across all patients, or did lesion location change the picture?
So the structural efficacy was beneficial throughout for patients with all sorts of locations and focality and reticular pseudodrusen presence or absence. Where we did see a difference, and this was a pre-specified analysis, it was the visual acuity benefit was seen in the extrafoveal lesions as we hypothesized, because in the subfoveal lesions, the photoreceptors are either already atrophied or quite damaged. So we expected to see preservation or visual benefit if the drug was successful in extrafoveal eyes. So it was precisely in that stratum that we saw the BCVA benefit.
Were you expecting these surprising results, or did any of it surprise you?
So this is the work of over 20 years by many different highly uh, capable and industrious individuals at the University of Kentucky, University of Virginia, and later at Inflammasome Therapeutics, where we showed in multiple papers published in Nature, Science, and Cell that K8 and similar molecules have a tremendous reduction of RPE and photoreceptor damage across multiple animal models of geographic atrophy. Geographic atrophy, as we all know, is a disease that's multifactorial. It's not just one thing, which is one of the lessons we have learned from the complement inhibitor saga, that even when it does work, which is in just 2 out of 15 trials, the effect is very modest. On the other hand, a drug like K8 that blocks a final common pathway, which is inflammasome activation, which is downstream of complement, amyloid beta, iron, retrotransposons, oxidized lipids, all these things converge on inflammasomes, and that's why we expected and we predicted from our animal studies that K8 would indeed be substantially more effective than what we have seen to date.
Let's go back to the safety. It has been a sticking point for the approved GA therapies. What did you see on that front over the 6 months, and what were you watching more closely?
Yeah, so the first thing is the only adverse events that were noted that were drug attributable were subconjunctival hemorrhage, and that's primarily an injection-related event, and the subconjunctival lidocaine that was used in some patients. There were no choroidal neovascularization events over the 6 months, no intraocular inflammation as well, and no vasculitis or optic neuropathy, and no IOP increases of note. So all in all, we couldn't have asked for a better safety picture, especially as you point out with the two approved drugs, there's anywhere from 10 to 12% CNV, 4 to 5% IOI, and so on.
We're a couple of years after the first GA therapies are approved. A lot of retina specialists are still on the fence about whether they want to treat this disease at all. From where you sit, what might be driving that hesitation?
Yeah, I think the hesitation, which you correctly point out, is driven primarily by the stark contrast to our wet AMD experience. There, patients actually can see, a large fraction of them can see visual improvement when they come in month after month. Here, with the two approved drugs, there's no VA benefit that's seen, and the lesion continues to grow, and it's hard, if not impossible, to ascertain any benefit from a month-to-month visit. And that's why there's a lot of attrition in real world practice as well. The other thing is, of course, the safety concerns for drugs that have, according to their FDA label, very modest structural efficacy and absent visual benefit. It's hard to make the case. And I think that's in large part why you only have 10 to 15% market penetration at best for the two approved drugs.
How much of the future of GA treatment do you think comes down to the drug itself versus how the drug gets delivered and how often?
So, apart from the structure and functional efficacy benefits, as well as the safety profile differences that we have seen with K8, there are three other parameters or dimensions on which we think it is a transformational advance. So, safety, efficacy, but also durability. So, in contrast to the two approved drugs, which are injected every month, K8 is injected once every 3 months. So, you already have a 70% or so reduction in treatment burden every year. The fourth dimension on which it benefits is the fact that it doesn't require any cold chain logistics. It's a small molecule with a very extended tabletop half-life, unlike the two approved drugs that require refrigeration, and that often limits accessibility to rural areas or satellite clinics, and certainly requires great outlays in capital on the part of practices from alarm monitoring and cold chain. And finally, ours comes in a K8 comes in a preloaded injector. So no need to watch a 9-minute video and drop vials and potentially break sterility and potentially have increased risk of endophthalmitis or intraocular inflammation. So on all these five parameters, we're seeing clear differentiation of K8 compared to the current landscape.
If you could get every retina specialist to unlearn one thing about GA, what would it be?
That it is a death knell for vision, that it's a disease that cannot be stopped. We see now with K8, we can see tremendous reduction in growth rates, and that too right out of the gate. We saw reductions within the first 3 months. So not only does K8 work well, it also works right out of the gate. So this is a disease in which we now have a different kind of paradigm that we can potentially think about, assuming the phase 3 results bear out what we saw in our very robust phase 2 data set.